My name is Margaret Lentz. I am a program manager at Department of Energy, Office of Science, Advanced Scientific Computing Research. And I'm glad to start by introducing Danilo Pérez Jr. from New York University, whose field is neuroscience. It's my field too, I like it. Yes. Advisor Christine Constantinople, and his practicum was at Argonne National Laboratory in 2022. And the title of his talk is Hierarchical Kalman Filters Reveals Multi-Timescale Neural Dynamics in Orbital Frontal Cortex. Danilo?
Thank you very much for that introduction. And though I do feel well accompanied by a fellow neuroscientist, I guess this title doesn't exactly point towards why we would care about these multi-timescale neural dynamics. So I wanted to offer a quick example of what type of phenomena we aim to understand as neuroscientists by using these tools. So to give an example of this, we have this group of people that seem to be in line waiting for a restaurant. And in a particularly restaurant-dense neighborhood, if you were to arrive at this line and see that your expected wait time is above 20, 25 minutes, you'd say, well, perhaps I'd be just best fit opening Google Maps, finding another restaurant, and just moving on over. However, if you are in a different neighborhood, quite common if you aren't downtown in New York and you had to head somewhere else where you were more restaurant sparse, you'd probably stick around because the quality of food or exactly what you were looking for isn't exactly immediately available in your context. And this is what we mean by subjective value being dependent on context. In situations like these where Google Maps is readily available, we can answer this directly with complete information. However, more commonly, we face decision-making every single day of our lives without such complete sources of information. And that's what we aim to understand as neuroscientists, and we hope computational models help us extract this sort of information more usefully from our experiments. So in the Constantinople group, a task was designed to try to elucidate the same phenomena or similar phenomena in rodents with partially observable or incomplete information. And we call this the value-based temporal wagering paradigm. Quick explanation. Each trial starts with an initial engagement of the rat in a sense report that has been illuminated to indicate that a trial has begun. And upon engagement, they'll receive a cue that they've been trained to recognize and associate with a particular reward offer, 5, 10, 20, 40, or 80 microliters of water. They've been water restricted, so they're highly incentivized to participate. Upon receiving the cue, one of the side ports will turn on, indicating where they can retrieve the reward upon the light turning off, or if the light doesn't turn off, they can visit the opposite port to opt out. What's controlling the timing of that light turning off is an exponentially decaying distribution, so they are very hard set in being able to predict exactly when that light is going to turn off. And furthermore, 15 to 20% of trials are actually catch trials, forcing them to opt out and receive a new offer. So what we would immediately expect from such a paradigm is that with increasing reward offer values, if the rodent can recognize what's being presented, they should be more willing to wait for larger rewards and less willing to wait for smaller rewards. And effectively, that's what we get. Upon constructing this paradigm, one manipulation we can make is to structure the rewards into blocks. What we mean by that, in a mixed context, you'll see a distribution of all 5 rewards. However, in a low context, we can restrict that to just the low reward offers. And in a high reward context, restrict it to just the high reward offers. And rats, as I show here, we have about 240 rats executing this paradigm. And we've collected their data for modulate their wait times based on the context. In low context, they'll increase the amount of time they're willing to wait for the 20 microliter offer, which is shared across all 3 contexts, while in high context, they'll reduce it. This is reflective of the fact that in low context, 20 microliters becomes the best available offer, and they'll be incentivized to wait further for it, whilst in high context, it actually becomes the least valuable offer. Given this behavioral readout, in neuroscience, obviously, we're interested in identifying where in the brain these behaviors might be driven. And so in the literature, it's been well established that OFC, or orbitofrontal cortex, is involved in valuation, particularly in terms of valuation future rewards and computing quantities that might be relevant to it. And a graduate student in our lab prior to my arrival executed an experiment where she took control rats who exhibited the behavior I just exemplified across 200 rats in a more restricted example, because now we aren't just running them in behavioral rigs, but actually executing experiments. And what she did is she injected into the orbitofrontal cortex a substance known as mucimol, which we can just think of as essentially turning off the OFC. It mimics the inhibitory activity by binding to GABA receptors. And the animals actually are still able to modulate their wait times based on the immediate offers that are presented. But what is affected is their context sensitivity. So this gave us a early data point that, in fact, orbitofrontal cortex does play into valuation, but in a more subtle way than what one might expect. So moving forward on this new information, Shannon also prepared experiments to be able to implant the rodents and now actually record the orbitofrontal cortex activity as the rodents were participating in our paradigm. So as you can see, we have a number of rigs, and that's what facilitated us getting 200 freely behaving rats and participating in our paradigm that are automated. However, injecting these probes that are highly sensitive is a much more sensitive process, and we are restricted in the amount of rodents that can be collected. However, what we do get are very high dense recordings of simultaneously more than dozens, can be up to hundreds of neurons being simultaneously recorded. And when we inspect these traces that we record from these neurons in the OFC, we can find that at 20 microliters, dependent on which block the animals are found, we can find neurons that seem to encode or modulate their firing rate according to what block they're present in. However, we can also still identify animals that within a particular context also modulate their firing rates in response to the offers that they're being presented. And so this was a key clue that we should leverage some of the statistical properties that in the literature are well established exist in neural recordings. One in particular, borrowing this illustration from Zheng and colleagues, is the fact that simultaneously recorded neuron activity is not independent, right? So if we had n number of neurons, in this case exemplified by 3, it is quite common to find that they're correlated and in fact inhabit a subspace, in this case n of 2, to exemplify this dynamic. So in our highly, highly dense recordings of dozens and dozens of neurons, there should be a subspace in which these neurons are encoding these distinct variables. And that's going to guide our model design process. So as I mentioned, there seems to be 2 key timescales. There's a fast within timescale, recognition of reward, but also a multi-trial accumulation of information that we believe represents a slow dynamic. So with our spikes that we're recording from the orbitofrontal cortex, we believe these to be representative samples of a fast dynamic process that evolve on top of a slow dynamic. And a natural way to model this is then to make a one-to-one correspondence in what we call these fast dynamics within trials that our spikes produce are readouts of, and the slow latent that drives that fast latent evolving on a slower resolution on a trial-to-trial basis. Mathematical formulation of this process might seem familiar to you guys and is kind of given away in our title, is a Kalman filter with the addition of this slow latent process that drives an additional input into the fast process. The math is essentially worked out, and latent inference follows the standard predict update nature of the Kalman filter, and parameters can be learned using expectation maximization, which we've kind of extensively talked about numerical optimization methods here, so you guys can probably predict that this is where high performance computing comes in. Given the data volume, right, we have dozens of sessions, about an hour length each, 20 to 80 simultaneously recorded neurons per session, fixed latent dimensionalities. We needed over 30 seeds fit on each session. So thanks to the access to the green supercomputer over at the high performance computing facilities of NYU, we were able to execute these computations and extensively optimize the model as well as test the hypotheses of interest. And now I'll walk you guys through some of the results that the hierarchical Kalman filter were able to highlight. First off, right, we validate the fact that the model produces sensible readouts on held out test data. So what we do is we fit the model, hold out a particular neuron, infer the latent space dynamics, and then project it using the learned parameters on neurons that aren't included in the inference procedure. And we can see that we obtain satisfactory reconstructions of that held out test data. Now, with this essential validation step reproduced across distinct neurons, we move towards the actual questions of interest of what is being represented in the fast latent dynamics and what is being represented in the slow latent dynamics. As I mentioned, neural recordings are quite noisy, right? And from a signal processing perspective, one would expect that if we have multiple neurons that we can identify encoding particular rewards, what will happen in the fast latent space is that a dimension should emerge where a much cleaner representation of reward can be read out. And effectively, this is what we get in the fast latent, indicative of the fact that the fast latent is encoding single trial dynamics. Moving above towards the slow latent process, we mentioned that in the wait times, there is a modulation that suggests that context should be reflected in the trial-to-trial updates. Effectively, when inspecting a single animal, we see that, in fact, the slow latent value is essentially encoding just context, whilst reward is essentially ignored. Now, this is just a single animal out here on the right side, but this actually generalizes across sessions of what we call expert animals, which are animals that have extensive exposure to this block dynamic and have participated in our task. Interestingly, it's attenuated in naive animals, so these are animals that are just gaining their first exposure to the blocks, and we wanted to inspect this even further because a further description of behavioral modulation on a trial-to-trial basis kind of suggests that in the mixed block, since you're observing all 5 reward offers, you really can't detect changes into low or high blocks. You simply have to modulate, and we presented evidence of this in a recent Nature Communications paper based on the recent statistics. So you're not going to receive confirmatory evidence that you've transitioned from mixed to low as you would in the opposite direction. So if you were only observing low offers, moving into a mixed block is almost explicit because after a sequence of low rewards, you would have a 40 or an 80 microliter offer, and that would tip you off to the transition. But the reverse transition is quite slow and has to reflect a belief updating process. We wanted to see if such a thing emerges in the slow latent, and effectively, we do see evidence of this, suggesting that the slow latent, which is computed directly off of the neural recordings and has no knowledge of the reward offers or block transition structure, does seem to be represented in the neural recordings. However, as I mentioned, there is a reverse process, which is explicit, and we wanted to see what distinctions might occur in the experts. So as we can see this incongruent trial that I mentioned, in a low block, beginning to observe in a mixed block 40 or 80 microliters, and a high block receiving a low offer of 5 or 10 microliters, seems to mark a sharp boundary that the slow latent captures in expert animals. This is seemingly not present in naive animals, suggesting that any sort of early modulations that we're seeing that previously mentioned were attenuated emerge from a distinct sort of calculation that the naive animals execute in terms of their navigation of our task. So slow latent belief representation is actually driven by experience and suggests that there's further inspection that the HKF can help us realize to understand the learning of these task dynamics in the animal. So given that, I'd like to recapitulate our results. The HKF was constructed and cross-validated, producing interpretable latent dynamics that help us pin down the nature of the neural computations in OFC, particularly the fast latent dynamics reflect within trial dynamics. Slow latents reflect the hidden state that the animals find themselves navigating, and the slow latent dynamics are consistent with belief updating, reflective of the information that they receive on a trial-to-trial basis. With that, I'd like to acknowledge both the Constantinople Lab and the Savin Lab, of which I am being co-mentored, and I'd like to thank particularly my committee members and the Department of Energy Computational Sciences Graduate Fellowship for the opportunity to pursue cutting-edge high-performance computing research in neuroscience.